BCR, basically the advantage of this binding site is it can bind to the antigen directly. I told you that it does not require any kind of antigen presentation, the B cell. It has the capacity to bind antigens. The T cell cannot see antigen unless it is presented in the context of MHC, and it has to be broken down into peptides. So what people did is they used this specificity. So this is what I have shown you, that it can bind these protein antigens, pathogens, and it can give them signal. And in case of T cell receptor, it can interact with the APC via the T cell receptor. So what people did is, so okay, let's say why cars, okay. So they took the best of both worlds. So one of the problems with the T cell receptor is that it cannot bind antigens, but the B cell can bind antigens. So they took the B cell part of the binding part of the B cell, but what they did is combined it with the T cell receptor, because the B cells cannot kill the tumor cells. It can only make the antibody. It does not have the property of killing the cell. So they took the signal transduction part of the receptor of the T cells and the antigen binding part from the B cell. So making a chimeric receptor. So taking the best of both worlds. So the antigen can bind to the B cell receptor, and the signal which is given is same as any like a T cell signal, which will result in a cytotoxic T cell response. So that's why it is known, because it's a chimera, right? So that's why it's known as a chimeric antigen receptor. So it's a form of an adoptive T cell therapy. So the receptors, this is not a common thing which is found. So the receptors have to be synthetically engineered for the T and B cell receptors have to be synthetically engineered to overcome the immune tolerance and tumor evasion. So the advantage is that it can target the molecules in their native conformation, because it does not, even though it is a T cell, it has the B cell receptors, so there is the broken down into peptides. So the whole protein can be recognized. And you don't need an antigen presenting, because B cell can bind, the receptor can bind the antigen directly. So just that whole, the T cell which you have made, the chimeric one, is enough. You don't need any other cell besides it. So this is an example of where they have made an CAR T cell against CD20, basically against the B cell. CD20 is a receptor which is expressed on all B cells. So they have taken the binding region of this, taken this CD3 region or the signaling region of the T cells, and made this CAR T cells, which can bind now to CD20, because this antibody was against CD20 and transduce this signal. Now, if you put these T cells back, it can kill all CD20 expressing tumor cells. So it can lead to tumor eradication. Basically, later this part is more or less the same. The only thing is that now it is able to recognize this tumor because of the CD20, and the killing mechanism is still the same, because it is basically a CD8 T cell, which is going to produce granzyme and perforin and do fast cell lysis, and this will lead to death of tumor cells and tumor eradication. So now many more different kinds, like people are putting more SCFV or single chain fragments. These are antibody engineering techniques, which were already used for other things. Now they're being used for this kind of a thing. The only thing which you have to do is, in all cases, you have to make the CAR T cell specific for that any molecule on that tumor. It's not that you made this CD20, it will work for another tumor, because this CAR T cell which you have made is against CD20. So unless the tumor is specific, is expressing CD20, it won't work. So you have to make for each tumor separate one. So far, So this is the whole process of CAR T cells. So basically, again, it is kind of a tailored therapy. So that's why it's also an expensive therapy. So blood is taken from the patients, then you have to take out T cells, are sent to the lab, right? In the lab, you have to do this, and now people are, one of the things which happens is, now that it has shown success and it has been given approval by the FDA, because it's a very different kind of therapy. If you see, you're doing transfection with viruses and then putting back into patients. So all this has never been tried. So people are, the companies or the labs which are working on it didn't know that such therapy will be approved or not. Now that it is approved, now they know, okay, if we put money in it, it may give us something back. So
No.
more is going to be tried. The other thing is, yeah.
Ma'am, do you believe that this B cell tumor and all coming from stem cell?
It
Because
depends.
because hematopoietic cells are giving rise to all different types of cells.
Mm-hmm.
So once it is differentiated, that's all. It will destined to death after function.
The stem cell?
No, the differentiated one.
Okay, yeah.
Okay, but the source of tumor is the root is inside the bone marrow only. So why do company invest just for one shot?
No. So it can be both ways. It can also be one B cell clone which is there already differentiated, which has multiplied to such an extent, and that is seen in many B cell lymphomas, that
The tumor may rise from non-stem cell also.
non-stem cell also, because
Which means already B cells are formed. From there, it is rising.
Yeah, it can come from the bone marrow cell also, but many a times you find that certain clones of B cells have multiplied. So then it is not coming from the stem cell. It is the B cells which are already differentiated have certainly acquired the capacity to proliferate because of any kind of transformation.
Because no, ma'am, this colon cancer and all. See, every 4 days once the cells are dying, getting
new
shedding.
cells are coming.
If cancer is forming, it should undergo cell death, and then it should be.
So one of the properties of cancer cell is it doesn't die, right? That's one of the properties, that it is immortal. It doesn't die. It will proliferate. That is one of the that's why you're targeting all the proliferation and apoptotic pathways, because you want the cancer cells to die. If they are dying, no problem. Because they divide indefinitely, right?
I didn't understand this CAR T concept very clearly. You are telling, no, ma'am, we are targeting CD20. But why do our cell will target our protein only? That is like it is present in our
cell, but you're making this cell. It's not your cell, right? You're taking the B cell, which because you've made an antibody against CD20. So now that antibody has the binding region for CD20.
Oh, okay. From that they
are taking that. That's why I was saying that for other molecules, you have to first identify the molecule. It should be a cell surface molecule.
Then we should engineer every time.
Then you have to engineer every time. So it's not as simple. It's also a very highly tailored therapy. In fact, if you see, it requires more steps because it requires lentiviral transfection. Because this engineered receptor has to be put inside the cell so that the cell expresses it, right? And then you have to expand those cells before you put them back. So the region which, from the antibody, basically, it's basically a whole transfection mechanism to express the receptor on the T cell, expressing the B cell receptor. T cell expressing the B cell receptor is specific for your target of choice. In this case, it is CD20. Okay. CD20 was the first one which was tried. That's why you see mostly those examples. So it is found on B cell malignant cells, and these are CLLs, ALLs, etc., and expressed on early B cells but not on stem cells. This is important. This is talking about only CD20. Okay? Because you don't want to kill stem cells. If you're going to kill the stem cells, then the B cells cannot come back, the normal ones also. So you choose an antigen. So in this case, let's go back. if you, in this case, you are wanting to target B cell related cancers. Okay, so you want to get rid of B cells. So now you have chosen an antigen CD20 because it is expressed on all B cells, but it is not expressed on the stem cell. Because when you will give this therapy, it will target all B cells will go. That will include cancer cells plus normal cells. Because CD20 is specific, not specific for cancer B cells. It is expressed on all B cells. So all B cells are disappearing from your body. Because the stem cell is there, it will make new B cells. And because of that, because you have gotten rid of the cancer cells, chances are that most of the new B cells will not be cancerous.
Mutation is there in the stem cell only, then
this will not
work.
So then it is a different problem. Then it tells you that the mutation is a stem cell. But in most cases, like I told you, that it is few B cell clones which become expanded. So it is going to work in those kind of cancers because there is no change in the no change in the stem cell itself. It's the B cell which has already differentiated, come out, and you have 10 to power 8 B cells. Out of those, 10 have decided for whatever reason, whatever carcinogen, UV or whatever it is, they have become transformed and those have started to multiply.
How come in 10 days the normal cell became cancerous cell? It's not possible, no?
Say again, I didn't know.
The hematopoietic stem cells are giving B cells.
So why can't the B cell become transformed? What is stopping it?
No, ma'am, 1 or 2 mutation is not sufficient to.
But how do you know there's only 1 or 2 mutations? Why can't it be 8 mutations?
That's what, ma'am. In 10 days, how come so many mutations will?
But it could be 1 or 2 mutations, but the mutations which are really aggressive. It's not required. There's no rule that you have to have 10 mutations, then only the cell will be transformed to a cancer cell. It can be 1 mutation, it can be 2. There is no fixed number, right? That okay, once you have 5 mutation, now you will become cancerous.
Because
if you have 1, I will not become cancerous.
Because in our lab, no, ma'am, SV40.
Yeah.
In our lab, ma'am. In our lab, ma'am, SV40 we overexpress H-stat, but still it is not transforming. That is a.
But SV40 is very commonly used to immortalize the
correct,
cell.
but still it is not behaving like cancer only, ma'am.
So it all depends. So in B cells, it is mostly EBV which is used for transformation to make them immortalized. So depending on the cell type, the virus also changes which you want to make it. Yeah.
Because after differentiation, there is not much time for cell to die. But in short span of time, that I believe, ma'am, I believe that the cancer is coming from stem cell only. I believe like that.
No, that I am not ready to agree, because then these therapies should not work, right?
Maybe it is minimizing initially. After that, again they will come back with recurrence.
But in many patients there is no recurrence. So that tells you that there is no. It's not coming from a stem cell, right?
Yeah.
If there's no if it is 1 clone or few clones which have multiplied, and now that you have gotten rid of them, you are free of cancers. So it all depends on which stem cell he's talking about, at what stage of differentiation, right? Otherwise, you are going to have cancer. So.
And then, madam, on stem cell, do you the expression of CD20 will be seen?
No. See, this is what it says here. If you see that the reason it was chosen is it is expressed on early B cells and not stem cells. Because if it was expressed on stem cells also, then you are basically getting rid of the stem cells. So once the therapy is done, there's no stem cells, so no normal B cells can be made. So you cannot get back your normal cells also. So that's why you have to choose the target with a lot of thought. So that's why they chose CD20, because it will get rid of early B cells, because the transformation the cell could have become malignant at early B cell stage. There are different stages of B cell development. It can be at early B cell stage, middle B cell stage. So many, if we look at B cell development, there are at least 10 different stages which are there. So that's why this particular antigen CD20 was chosen, because it will get rid of early B cells as well as late B cells, but not affect the stem cells. CD19 is also a similar kind of molecule. It is, in fact, if you want to label your B cells with flow, and you want to say it is a B cell, you use an anti-CD19 antibody or a CD20. So it's a specific marker. So what are the advantages of CAR T cell therapy? That it is an HLA independent antigen recognition. So basically you don't need, so it is of universal application. So you don't need to worry about any kind of mismatch of MHC, that okay, it cannot be given to another. And this is active in both CD4 and CD8 T cells. Target and antigens can include any kind of antigens. It can be carbohydrate, protein, or glycolipids. Rapid generation, because now the techniques for this transformation are well set. So you can, like, people know how to do this 40 to 50% transfection efficiency, choosing of the right kind of T cell expressing the right kind of receptor, and then how to expand them and putting them back. Minimal risk of autoimmunity or GVHD because it is targeting a particular antigen. It's not removing the brakes on the immune system, right? It's a living drug, or a single infusion is supposed to be sufficient so far, because it's only been tried in B-cell lymphomas. So this is just to show you this. This is one of the articles. Maybe first I'll show you the video and then we will talk about, no, I can do that. No. And I have to do the duplication, right? Let me, yeah, it's coming. Oh, it's coming back.
T cells are normal cells of the immune system that help to fight off infections. T cells can react and kill cancer cells in the right time. CAR T cell therapy is very unique and different than any other treatment that we've had. It's a treatment where we take the patient's own T cells and we modify them in a laboratory so that they can recognize and react against their cancer. The patient undergoes apheresis, which is the process of removing the T cells. We modify them in a laboratory to express receptors to better recognize cancer. Those T cells are then expanded and grown to large numbers. We reinfuse those modified T cells back into the patient through an IV. The CAR T cells that are generated all have a similar receptor on them, and this receptor recognizes a cancer cell. Patients can get flu-like symptoms or neurologic symptoms from the treatment. Right now, we're giving patients CAR T cell therapy in the hospital because their symptoms can become very severe. We're currently treating childhood and young adult acute lymphoblastic leukemia. In addition, we're treating patients with non-Hodgkin's lymphoma, particularly diffuse large B-cell lymphoma. MD Anderson researchers are working to expand application of CAR T cells to other cancers, including solid tumors.
I'll show you the other video also, and then we will move so that it's easier that way.
Chimeric antigen receptor therapy, or CAR T cell therapy, is an emerging immunotherapy designed specifically to target a patient's cancer cells. To date, the most successful of these therapies, CD19 CAR therapy, targets B cells in leukemia and lymphoma. A patient begins treatment by undergoing apheresis to extract normal T cells. In the laboratory, a modified virus, either a lentivirus or a retrovirus, transfers targeted genetic information into the T cells where it is incorporated into the genome. Newly synthesized CD19 CAR proteins are then expressed on the T cell surface. The altered T cells are expanded in vitro and delivered to the patient by infusion. In the body, the engineered receptors bind to specific antigens expressed by the target cancer cells, killing them. With hundreds of trials currently being conducted worldwide, Engineered cell therapies, like CAR T cell therapy, have the potential to transform treatment for cancer and other diseases.
So these are some other diseases which people think, because not only in cancer, but in other diseases, especially I've seen a recent report. We'll go back to this again in fibrosis, where they're using this similar kind of therapy, at least in, I think it has been done in mice, where they've targeted this fibroid cells, making CAR T cells specific for that, and it can get rid of the the cells which have become fibroid. And so one in cancer therapy, now you can think how you can engineer CAR T cells for other diseases, maybe where there's a single antigen which you know of, where a certain particular cell type is a problem. And if you can target that particular cell type, then you can get rid of that. So it's simply that, when something becomes successful or shows success, and the other major advantage, as I said, is that it has been approved in the clinics by the FDA. It is being given to patients. So clinical trials and everything has been done. And despite the toxicities and other things, the benefits are supposed to outweigh the problems. So then you will see a lot more of such therapies coming, because first approval is always more difficult than the future ones, simply. So here is this is a very, this is a paper where this was the first paper, I think, which showed that chemotherapy refractory. So it has been tried in patients where chemotherapy is no longer working, right? It's a large B-cell lymphoma. So in such patients, when other drugs are not working, and if you come up with a drug which can now do something, because basically then there is nothing left to be given to these patients. So then it becomes very important to find, that you have a therapy which can now be successful. So this is just showing you that this is a complete remission of chemo. This paper which I showed you, and this is what they had shown in this paper, that 23 months up, this is before treatment, this is also before treatment. You can say this tumor is growing, and after treatment, it has disappeared completely. This is 5 months after treatment, 9 months, and by 23 months, there is nothing there. Okay, so it is so successful that by PET and CT that it has completely gotten rid of your B-cell lymphoma. So now the next few slides, I'm trying to, what I'm simply showing you this, now I've already shown you what happens, right, with the video. So this is redundant at this point, but the next this slide is taken from a new article in Nature, and in this they have shown results of 4 patients who have, these are all pediatric and young adult patients who have relapsed or refractory B-cell lymphoma, BALL, right? And here, in all cases, all 4 patients have some differences or the other. All have failed whatever therapy has been given to them. So if you see, these are all different kinds of therapies which have been tried. So this is SCT or stem cell transplant. And still, basically, what I'm trying to say in the CAR T cell therapy is the green round thing. So all other, these are all patients which have the same disease, but exhibit different, have different types of the same disease, different kind of therapies tried, and they all have become resistant to it by this time. So after this, the CAR T cell therapy was given in all 4 of these patients, and so far, all of them are showing very, very good results. None of them have had a recurrence of cancer. This is about 2 years, 3 years. It's not that long, because the therapy itself is so new that there is you don't have that long-term follow-up reports so far. So this is what basically you do, that you keep them on maintenance. If there is a relapsed disease, you do it again. So okay, I think so one of the major complications of CAR T cell therapy is it is like when you have a lot of high level of immune response, especially in flu-like and other in flu infections and other infections, when you have, then a lot of cytokines are produced. Because when immune systems are active, and when if there's too much of this cytokine release, it causes something which is known as cytokine release syndrome, which is a little problematic. And this is usually typically within 5 days of CAR T infusion. That's what they have realized. And because this cytokine release will occur early only, right? And if you look at the CRP protein, it is the best predictor. If this is going up, that means this patient may have this cytokine release syndrome. Because exponential T cell, because these T cells are so many high in number and they are dividing, it leads to high production of these cytokines. Because T cells, when they are activated, they will produce these cytokines. You saw that T helper cells, anytime an immune cell is activated, it will produce certain cytokines. Especially T cells will produce interferon gamma or IL-17 or something like that. But in this case, they are producing more of interferon gamma. And which the interferon gamma I mentioned can act on macrophage because it is a macrophage activating factor. So macrophages are activated. So it is more like a sepsis kind of a situation, but not to that level, okay? But it can lead to that if it is not treated. So what they have, the treatment for this is you give this anti-IL-6 antibodies, which are already approved for clinic. The other thing which you do is give steroids, which are basically immunosuppressive drugs. So it's a short-term problem which is there, okay? But not, at least so far this has been seen that if you give these therapies, then you can keep the patient. So you have to give it like for a week or so, patient goes back to normal, but the cancer does not come back. So it's a short term. So you have to do it in the hospital setting so far. You can't send them home, like give them CAR T cells and send them home. You have to keep a watch on for all these complications. So B cell aplasia, this is simply because, as I said, in this particular CAR T cell therapy, you're getting rid of all B cells, right? So there are no B cells producing antibodies to fight fight infections. So people may be more prone to infections. So what you can do is you can give them intravenous immunoglobulin, similar treatment to what was given to primary immunodeficient patients who genetically don't have functional B cells or cannot make antibodies. So it's a similar situation, so you can take care of by doing this. So this is another problem which is seen. So it doesn't seem to be that common, but 6 in 30 patients have been show signs of this. But it seems to be self-limiting, like it occurs and then goes away. There are no long-term complications. And CAR T cells seem to be present in CSF of all patients. So I'm not saying that it is like a wonder drug, but so far the benefits seem to outweigh the complications. What will happen in the future is something which I can't predict. So what are the problems associated with CAR T cell therapy? Feasibility, which I've been saying again, it is a tailored therapy, and as I said, it involves more steps, so it's a very expensive therapy. Who is going to pay for it? Okay. Physicians need to be specially trained for it, in the sense they need to be aware of all the symptoms. They need to know what to look out for. Cytokine syndrome will be there, so they need to order the tests at the right time to see that the patient doesn't suffer from these complications. And relapse of tumor can come back. Depends, as I said, where that mutation is. So defining best CAR T cells. So there is, some people argue that retroviral transfection is better than lentiviral. This is a matter of choice of the lab. Okay, I don't know which one is better. So So now what people are thinking is, can we add immune checkpoint therapy to these T cells so that you can make them better? Sorry. Okay. And so far, it has only been, see, all these B cell tumors and T cell, which for which it has been tried, these are all non-solid tumors. Like B cells are in circulation. So far it has shown effect in non-solid tumors because you do an infusion, cells are right there, so they can see the target very easily. The problem is more in the solid tumors, where you give the cells, now they have to reach the organ where it is, go inside the tumor, because it can maybe attack the cells which are outside, but it should be able to penetrate the tumor also to kill other cells. That's where the major problem is coming. But this is the first step, right? That at least you are able to kill liquid tumors. Now what people are trying to engineer is maybe tissue degrading enzymes like hyaluron solid tumor, it can degrade the connective tissue and penetrate. A lot of different approaches are being tried, how to penetrate, okay? But still very early. So far, this is the only one which is successful. So this, now we are moving away from CAR T cell, and this I'm simply showing you that, I had mentioned that Th17 cells are opposite of Tregs. So many people, especially in melanoma, I think it has been shown that if you take out the tumor infiltrating lymphocytes, melanoma is your skin tumor. And if you expand those IL-17 cells, it seems to be it seems to serve better. And if you have more of IL-17 cells, you have more chances of tumor regression, because these are highly inflammatory cells. So if you have a highly inflammatory immune response, then again, there's not going to be immunosuppression. If there is an active immune response against tumor, you are probably going to be able to fight it better. So I can go through this. Okay, let me. So here, this is the tumor. It's making all these antigens, right? Tumor associated antigens, because these could be some of the molecules which are overexpressed or which have become transformed a little. These can be any kind of these tumor associated antigens, and they are taken up by dendritic cells, which are now activating, priming CD8 cells, and you're getting CTLs. And similarly, if these DCs are making a lot of IL-23, so this is a key molecule basically. If DCs make IL-23, the kind of T cell response you will get is going to be IL-17 kind of a thing. And IL-17 can be wait, let me think. So far, all this what has happened is good, right? So the IL-17 cells is going back, and then it is activating NK cells, CD8 cells, and all these things are going to attack the tumor and kill it. But the other problem which is associated with this is VEGF, I think. That Th17 cells also secrete this VEGF and other factors which enhance angiogenesis. In this case, it is maybe beneficial for the tumor, but it is also beneficial in the sense other immune cells can come into the tumor. And here it is beneficial because the whole environment is inflammatory. Angiogenesis is a problem when the whole tumor environment is suppressive, and angiogenesis is occurring, so all the nutrients are being taken up by the tumor. But if it is not immunosuppressive, then immune cells are able to come. They are not suppressed. They will also help in fighting off the tumor. So this is just one slide to show that how inflammation, if you enhance inflammation in the tumor, it can be successful. So this is another approach which some people are trying to deliver drugs via nanotechnology. So this is one paper which it's a very recent paper published in January of this year. And here what they have done is they have taken a lipid-based nanocapsule, and they have taken this drug gemcitabine, and it can deplete these myeloid monocyte, myeloid-derived suppressor cells. What I mentioned, that they are like T regulatory cells. Their also major function is to suppress immune cell function. So basically, they are using these lipid nanoparticles to deliver this drug, which will go there and kill these myeloid-derived suppressor cells. So basically, they are making the preconditioning the tumor microenvironment and remove now if you give the immune checkpoint inhibitor or some other therapy, it is going to be more successful. So in a way, it's making, people nowadays are thinking of many ways how to improve therapy, even for immune checkpoints. Maybe you should already precondition the tumor microenvironment, that you give something that it is already becoming a little more activated. Now, if you give immune checkpoint therapy, it is going to be more successful. The other approach which is being tried is in some tumors. So one of the things with the immune checkpoint inhibitors is not all tumors express this, PDL1, PD1 or CTLA4. So what do you do in this, especially PD1? What do you do in these tumors? This therapy will not be successful. So far, it has shown very good effect in non-small, mostly lung cancers quite a bit, and now approved for, I think, other ones, maybe breast also, but started with lung cancer. But I think colon cancer or one of the other cancer, the cells don't express PD1. So now people are also working on different ways how to enhance expression of PD1 on tumors, so that now you can use this therapy, because this is an already approved therapy, right? So one of the cytokines which is known to do this is interferon gamma. So interferon gamma is now being given to for a different reason than other, okay, it is also a tumor stimulant. It may help at least do a little bit better in removing tolerance from the tumor. Side by side, it may also lead to expression of PD1, because the immune checkpoint inhibitors are not given like this. First, a whole battery of tests are done to see if this expression is there, and now people are coming up with certain modalities or certain signature which will tell them that, okay, these patients are going to be successful if given the immune checkpoint therapy, while these may not show an effect of this kind of a therapy. And that is known as, now you can say, that's a new era of cancer diagnostics. It's like personalized medicine. And that I'm going to talk about tomorrow, a whole lecture of all these new approaches which are being tried. Because earlier, any patient would come, you would give a combination of 2-3 drugs, Taxol, and now that is no longer an option. And that's why it was irrespective of what you have, you will be given these chemotherapeutic regimens. Or if it is a lung cancer, this is the chemotherapy you should be given. Now they're realizing that there is no need to give so many of these drugs, because many of them, if you're not showing a change in that pathway, it is of no use. Instead, it causes multiple problems. So doing this personalized medicine, there are many advantages. It is expensive, definitely, because you have to look at each patient, but the patient suffers less side effects. On the whole, it is less expensive, because you don't give the chemotherapy. It is more expensive in the beginning to maybe sequence the tumor or do other things, but later on, you don't give that many cycles of chemotherapy. You don't use those chemotherapeutic regimens. So ultimately, people, at least in the insurance companies, have realized that it is cheaper to do the personalized medicine than continue with the others. So that is becoming the norm. So let's get back to this in a little bit. So this is a, yeah.
Like a solid tumor, this B lymphoma will cause metastasis?
B lymphoma is already in circulation, so it's kind of
okay.
metastasis.
So it will not go and sit any organ and cause metastasis.
Not really. Even if the B cells will go there, few B cells will go there, and also the T cells can also go there, right? Most of them are in. It targets. It's solid tumor is where you have a mass of cell in a particular area which is sitting there and growing. B cells will not attach to something and make a mass like that.
So it doesn't have
It
adherence
may
property.
go, yeah, because B cells are not, their function is to secrete antibodies, go to the place of infection and move around, right? So they don't need to sit in one place and adhere to something and make a mass out of it, right? Generally in solid tumors, what happens is our epithelial cell, say in the kidney or breast, has become transformed. And that is dividing. And that is in that place and making a tumor. It's not that cell is coming from somewhere and attaching there and then making a tumor. That happens during metastasis for sure. But then metastasis is so now there is also a lot of theories that when cells metastasize, they take along with them all these cancer associated fibroblasts and cells of the tumor microenvironment, which allow them to survive this journey during metastasis. So they take their own, like a own survival mechanism along with them, and then they go and seed into a different place where they already come with their own survival factors, cells which will help them survive, conditions which will help them.
It will go like a cluster.
Like a cluster. Yeah. So, and that is what some people are using now for diagnostic, like the circulating tumor cells, which they call CTCs, because they have to travel via the blood. So if you can get what stage it is, because there's a lot of interest in finding out diagnostics which can be done in the blood, because which are non, it's, so anything you can detect in the blood is considered kind of non-invasive, because taking out blood is the most easiest of things. For biopsies and other things, it does, either it's a small surgery. Okay, in some cases you can do a needle biopsy. It depends where the tumor is located, right? But it still requires invasion. And sometimes people believe that the disturbance of the tumor is not good. So there are many, many different things, which
Cyst form, if it is breaking, then it will spill. And
it is going to spill. So there is a lot of different problems and other things which are associated with. But still, I think we have made a very good progress now that we have some therapies which are showing promise. And in all these, in many of the immune checkpoint therapies and also CAR T cell therapies, at least whatever time it has gone, many of the patients don't show any sign of relapse, which is very encouraging for them. And also, it also enhances it. See, I also believe that it gives patients hope. And once the mindset of the patient is changed, I think that also helps them recover better. I think it adds a more of a positive thinking direction, whatever you can say. Once you are depressed, your cells are also depressed. And once you are happy, your cells are also happy. So they will try to do better.
We can inject happy hormone then.
That is chemically induced happiness. So maybe it doesn't work as well. Mind doesn't take it in the same way. So I think this is my second last slide. So we will see. So in this, okay, I've forgotten what I was looking at. So this is a EG7 ova lymphoma bearing mice, which was treated with this loaded nanoparticles. And then prior to the adoptive cell therapy, and then the survival was compared. And so this is a B16 melanoma is a mouse melanoma. So when this nanoparticles along with loaded with gemcitabine was given and you were preconditioned the tumor microenvironment, as you can see here, okay, I have to read, I forget. Because we were talking of so many different things, I completely forgot. So this is a mechanism where, so now first I have to tell you what it is, this blue thing which you are seeing. So Tumor cells, because they are proliferating so much, they utilize a lot of glucose. So you can do a CT scan where you give an analog of glucose known as FDG, which is a very an isotope of glucose, and it goes into cells which are utilizing a lot of energy. And if you do in a normal mice, you will see that brain is one place where it gets lighted up apart, and the pancreas. There are two places which light up the most, and that is one way of looking at tumor even in the patients, because this FDG is a nice, it has a very short half-life, so it goes out very fast. So it doesn't cause any problems in the patients, and it is approved. In fact, most CT scan people use it. So similar mechanisms people use for following mice, because the advantage of using such kind of PET CT scan for mice is you don't have to kill the mice each time to look at the size of the tumor. You can follow them. Some people have even done this, that they have made, in mice, if suppose it is a B16 melanoma, they have introduced GFP into this particular tumor cell, and now if it is injected into the mice to cause cancer, the tumor itself is fluorescent. And if you have an imaging, there's a whole body imaging kind of machine, it can show you where the tumor is and where it has metastasized also. So these are some techniques which are used to follow tumors in mice. So this is similar to that, is what I think. Okay. So here they are showing that lung metastasis is established in tumors. To read what, as I said, again I went to something else. So this is luminescence. So this is the third thing which I forgot to mention. So you can also make the tumor luminescent in like that, like you can introduce the luciferase gene. So then you can use a different machine basically to see the tumor. But all these are techniques which are now very much used in the mice. So mice received this injections of these immunoliposomes which contain this gemcitabine at day 0 and day 6. So this is day 0 and day 6. And then, so this is bioluminescence imaging of T cells. So it's showing how T cells have expanded in all these areas. So it's not of the tumor. That's why I got confused when I looked at it, because I thought, okay, the tumor seems to be much more in these mice where they have received, after treatment. It's more the T cells which are going in all places. So T cells are able to kill the tumor. That's why I thought, okay, maybe I'm looking at a wrong picture. So it's facilitating that the tumor is able to penetrate more areas and do their work when you are removing the myeloid-derived suppressor cells using this treatment. My reason for showing you this slide is that, we are talking about immunotherapy and other things, but nanotechnology is also something you can focus on, which is more easily doable in many cases, and less, how do I say, you don't require so many things that you need to take out T cells, you need to transfect them and then infuse back. Compared to that, this is much easier, right? If you can make something like this, a nanoparticle which can go and kill maybe Tregs in the tumor microenvironment, maybe in the whole body. It is similar to what you are doing with an immune checkpoint thing, right? Okay, you're removing the brakes, but that seems to work for tumors. It will cause some autoimmune diseases, but like in immune checkpoint inhibitors, you already know that so many diseases can be caused. But here also you can keep an eye on them, and it is a similar kind of situation. But this therapy is going to be much more easier to do, or cheaper also to do, right? So that's why I was showing you that think of other approaches. Okay, those are very fancy approaches, but very expensive also. But that gives you an idea that if you remove the brakes, you can get response. Now you have to come up with new ways how you can remove the breaks, right? That is the reason, I showed you this particular slide. And nanoparticles can definitely serve as vaccine platforms also. So you can use them for activate dendritic cells also. Maybe you can put some kind of activator molecule along with the antigen to be delivered to dendritic cells. Yesterday we were talking about, maybe yesterday or today, I don't remember, that okay, we are taking out dendritic cells, loading them with the antigen, putting them back. So if you can get an approach where you can take the antigen to the dendritic cells and load them inside the body, because if it goes into the dendritic cells and it is processed and presented, and so along with that, if you have an activator, which you are trying in other trials like the toll-like receptors, but you have them in the nanoparticle, then you're doing both. You're giving the tumor antigen and something to activate the dendritic cells. Now, these may be more effective in tumor because now you have an activated dendritic cell which can prime T cells and induce T cell immunity. I'm not saying it is going to be successful in the first time, but these are approaches which are more feasible for the long run, and maybe you should think about these things that develop these kind of approaches. So here they are saying that dual therapy of nanoparticles. So I could not change this because these are taken directly from the paper. So this is how they had presented that. So a dual therapy of nanoparticles in combination with immune check therapies. So basically, you're trying to get rid of some immunosuppressive cells. Side by side, you're also removing the brakes on the immune system. Basically, you can try many different combinations. It may be more successful. Here, what they have done is nanoparticles can modulate the tumor microenvironment. So to potentiate the efficacy of immunotherapies, this is what I was showing you, that they have removed the myeloid-derived suppressor cells and now find out that the T cell, if you now adoptively transfer activated T cells, they'll be more effective because there's nothing there to suppress their action. Because Tregs are one, myeloid-derived suppressor cells are others. There are many other factors in microenvironment. I think this should be probably the last. So these are, the four things which I mentioned, that you can use the nanotechnology to deliver cancer vaccines. So it can deliver antigens and or adjuvants to activate dendritic cells. So this is the first thing which I showed you, that the DC-based therapy, which you can do, you can try to do with the nanoparticles. Immune checkpoint blockade, you can also maybe deliver one of the immune checkpoint molecule and along with the drugs, maybe instead of giving them separately, to using the nanoparticles to improve therapy. Combination therapy is simply getting rid of certain cells and then creating a good environment for the drugs to act better. You can use it for cell tracking because if the nanoparticle contains any kind of a radioisotope or some kind of a molecule, you can track the cells. And adoptive T cell therapy is same manufacturing of T cells and adoptive therapies. These can enhance T cell cytotoxicity. I think this is probably the final slide which shows all the different kind of therapies which are there. This is CAR T cell therapy. Okay, we'll start with a monoclonal antibody conjugated to, toxins conjugated to monoclonal antibody can kill these cells. The other one which I mentioned, the dendritic cell-based therapy, infusing back into patient. Adoptively transferred T cells, you expand your T cells with IL-2, the tumor infiltrating lymphocytes, and give them back. They will kill the cancer. CAR T cell therapy, and this is the immune checkpoint inhibitors. And there is this video which will kind of do a recap.
Our immune system is equipped with cells that form an army that help our body defend itself and fight foreign invaders. Some of these immune cells, called T cells and B cells, are programmed to get rid of foreign invaders like viruses and bacteria. T cells release toxins against the foreign invaders, and B cells make antibodies to neutralize them. We have known for some time that our immune cells can recognize cancer cells as foreign invaders and attack them by secreting special factors or weapons. How can our immune cells do this? The organs of our body are composed of billions of cells. These cells contain thousands of proteins that act like worker bees and are responsible for the function of our cells. These proteins break into small parts called peptides. Some of these travel to the surface of our cells and are presented by molecules called MHC or HLA. T cells continuously scan the surface of these cells through the T cell receptor or TCR. The TCR works as a barcode scanning machine, and the peptides presented on the cells are unique barcodes. This function of the T cells is called surveillance. If T cells recognize these peptides as normal, then the T cells move on. But if the peptides come from foreign invader proteins, then the T cells get activated to attack the invader cell with weapons to eliminate it. The T cell secretes certain proteins and chemicals called cytokines and factors. These molecules punch holes into the cells that contain the invading organisms. Proteins are your body's basic building blocks. They allow your cells to divide, rest, and function properly. But sometimes these proteins get changed or mutated. For example, smoking leads to mutations that make abnormal proteins in our lung cells. Sometimes when these proteins are changed, they stop functioning normally. This makes the cell become cancerous by making it grow and divide to become cancer. These mutated proteins are also broken down into smaller pieces and are presented as peptides on the surface of the cancer cell. These abnormal peptides are recognized by the TCR as foreign and activate the T cells to kill the cancer cells. But if the immune system can recognize and eliminate cancer, then why do people develop cancer? Sometimes the immune system fails to do its job. The immune response against cancer may not be strong enough, or cancer cells may evade the immune system. Tumors, or a group of cancer cells, can build a defense network against our immune system. These defense mechanisms can either prevent the immune army from entering the tumor or can weaken and inhibit the T cells. For example, tumors can express attack molecules on their surface that bind to the T cells and inhibit their killing activity. These molecules are called checkpoints, and examples are PD-1 and CTLA-4. The tumor can also secrete proteins, factors, and cytokines that inhibit T cells from attacking and killing cancer cells, and can also turn the T cells into friends of the tumor. These cells can then deactivate other T cells. These traitor T cells are called T regulatory cells and are the enemy of the killer T cell army. The field of immunotherapy has shown great progress toward a cure for cancer after working at it for many years with some major recent breakthroughs. Immunotherapy is actually one of the most important revolutions in the history of medicine. What are the different types of immunotherapy? Immunotherapy functions in two primary ways: by enhancing the immune system and making them stronger, and by using drugs that help inhibit the suppressive immune environment of the tumors. One enhancement method is to create vaccines. Foreign antigens can be identified by identifying mutated proteins in the tumor. These mutated proteins, called neoantigens, can be given as a cancer vaccine. The cancer vaccine is developed to activate the patient's own T cells against the foreign antigens. Like the flu vaccine fights against the viruses, the tumor vaccine fights against the foreign tumor cells. Now that the T cells are trained by the neoantigen vaccine, they go around the body to find those specific mutated antigens and will locate them on the tumors. Another enhancement approach is adoptive T cell transfer, where T cells are taken out of the patient's body, grown in the laboratory, and educated to recognize cancer or even modified to become much stronger. These fighter cells are then transferred back into the patient. A third enhancement approach is using stimulating factors called cytokines. Cytokines are stimulating proteins like interleukin 2, 7, 12, and 15 that cause the T cells to significantly multiply and get stronger. A fourth enhancement approach uses agonist antibodies such as anti-OX40, anti-GITR, and anti-41BB, and others to cause the T cells to grow and strengthen. This allows the enhanced T cell to overwhelm the cancer cells. A second approach to immunotherapy. Strategies have been developed that knock down cancer defense mechanisms. There are antibodies that neutralize the inhibitory factors and cytokines, such as anti-IL-10 and anti-TGF-beta and IDO inhibitors. Some of these approaches have already been approved by the FDA, like anti-PD-1 and anti-CTLA-4. For example, patients in this clinical trial have melanoma, a kind of skin cancer. 50% of people with advanced melanoma die in 12 months, until recently. Patients were treated with antibodies against both CTLA-4 and PD-1. Growth of the melanoma tumor in different patients was recorded and plotted out in a graph. The growth lines show that targeting both CTLA-4 and PD-1 encourages T cells to fight cancer. With this combination approach, 70% of patients with advanced melanoma continue to survive more than 3 years. These therapies also have some side effects that need to be monitored very closely by the treating physician. Although the side effects are not very common, they could be life-threatening if not treated. These side effects are treatable, especially if discovered early, which include diarrhea because of inflammation in the intestine, include some endocrine diseases that lead to weakness, thyroid dysfunction, or diabetes, and other side effects that need to be discussed with the team. The future of immunotherapy is in combining different approaches to accomplish two goals: to create a trained army of cells to attack the cancer, and to break down the defenses of these cancers by unleashing your own immune defense against cancer to improve on the currently very impressive results that immunotherapy is already providing our patients.
Yes. So I think we finished with immunotherapy today, and we will talk about the cancer diagnostic and personalized medicine tomorrow, because that's a big lecture. So I think this is enough cancer for today. And thank you all for listening. So he was asking if when we are going to talk about the immunoassays. So what I was mentioning is that first we will continue with the cancer diagnostic tomorrow. That will take at least the whole of the morning. And in the afternoon, before it's starting, then I have mucosal immunity also and other things. So I was thinking maybe we can do some immunoassay and some other things depending on the time tomorrow afternoon, and then mucosal immunity on the last day, if that is fine. So maybe, so tomorrow, or I have not decided. Basically, there are two ways. Either we finish the mucosal immunity tomorrow afternoon, and the last day we keep like small 3 lectures, like a small ones. We start early and we do immunoassay, whatever is left. Some other, if somebody else wants something, otherwise I can talk a little bit about stem cells, even though it's not my area, but I have some, I can tell you a little bit about the background, how people are using. So I think that would be kind of enough. Or if there is any other area of immunology which is more interest to you all, then let me know. Because autoimmunity I can talk about, no problem. It's going to be more or less kind of similar to what I've been talking about for cancer. It's just the opposite in a way. But if there is a lot of interest, fine with me. I really don't. So you all can think and let me know tomorrow. Is that okay?